All right, everybody, let's get into this. We're going to talk about T cells and cell mediated immunity. So, in this case, we have two major cells, just like we talked about in the last video, the T cells and the B cells. So, these T cells are the attack cells. They are the most notorious cells that are going to go out and fight off the foreign cells by literally coming up to them and fighting cell to cell, right? So, in most cases, as you're going to see with the B cells and some of the T cells, they may just put something like an antibody into the bloodstream and have that do the fighting for it, or just engulf or consume the foreign pathogen. But in this case, these cells are going to attach directly to another cell to do something to it. So in this case, let's say this is our cell. And then let's say over here is not our cell, right? So the important thing is there has to be an antigen for it to attach to. And in this case, let's say our cell has this triangular antigen, and over here, the bad cell has the receiving end. So they will be able to connect. So it's kind of like a lock and a key. So our T cells are able to bind if they are able to bind, right? So there's a chance that the foreign pathogen has a different shaped antigen, right? So on the opposite side, maybe it looks like this. If it looked like this, that's a different story. It wouldn't work, right? But that's not the case. So if it's like a lock and a key. So if it does fit, what's going to happen is our cell can respond, right? And there's a variety of ways that our cell can respond. So in one case, it can release cytokines. So cytokines are going to enhance the cellular response to the antigens. So in this case, our cell, because it binds to it, once it binds, we're happy, right? And now we can release chemicals, these little dots, these little cytokines out to tell other T cells, hey, come on over here. We need more of you because we just found an invader, right? We attach to it and we know there's an invader here. So they're going to release the cytokines to tell other T cells to come to the area. Another thing that might happen, instead of the cytokines, maybe they release toxins. So in this case, these little purple dots, maybe they release toxins that will bind to the surface and destroy the pathogen. Another thing you can do is produce these growth inhibiting factors. So maybe in green. So maybe these growth inhibiting factors can attach and tell the bad or the pathogen it can no longer grow and it can no longer divide and stop it from uh, proliferating out and interfering with the body. And then something else we discussed earlier, it can release the interferon, which can go inside of the cell and stop it from reproducing, right? So that's more so if it's a virus. So if you have a cell, here's your good cell, and a virus gets into it. I'm going to use it in purple. So the virus gets in. Your cell can produce an interferon, which stops it from reproducing. So this is something you're going to see, like I said, in viruses and some tumor cells. So T cells have a variety of ways they can fight off the pathogens. And again, it is cell-to-cell contact. Here are different types of cytokines and what they do in the body. So there are multiple types of cytokines and they have a variety of functions. So activation. So here's another really important part of the T cells, and that is that they need to be activated, right? So T cell activation requires the presence of an antigen-bearing cell, such as a macrophage or a B cell that has already encountered the antigen. What does that mean? Okay, so let's take a step back. We didn't get this far before, but you might as well talk about it now. So let's say we have a B cell. And this B cell, or a macrophage, we could do a macrophage, right? The B cell is going to consume, let's say, a different bacteria, which I'll do right here in red, right? Once it consumes the bacteria, Notice that this bacteria has these little red lines, these little red antigens on the outside of it, right? So this bacteria has these little antigens. Once it's consumed, what the B cell is going to do is, I mean, it's kind of interesting to think about. If you ever played Super Smash Bros., here's an example. It's kind of like Kirby. So Kirby is going to suck up the character and then present part of that character on it. So just like that, the antigen from the pathogen is now displayed on the B cell, right? That's how you know that it had already encountered one of these pathogens. So in other words, it eats this, it gets consumed, it goes into it, and then it just displays parts of it on its own cell. So as twisted as that might sound, it's kind of like the same thing as someone who might take you know someone who wins a fight and then wears part of the person that they beat, right? So let's make it sound nicer. Let's say someone wins a boxing match and then starts to wear that other person's gloves, as a symbol of, yeah, I beat that person. All right, so that's a nicer way of looking at it. So here we go. The B cell, it's activated. The B cell is going to consume a pathogen and then it's going to present its antigens. To do that again, just to make sense of it, a different scenario. So let's say this is another good one. Here's another. Let's say they have these little squiggles. Once our guy ingests it, it is going to then present some of those antigens on itself. Okay, so this is what we're talking about when we're saying it already encountered it and presents the antigens. From here, right, we have different types of T cells that can be activated. We have the helper T cells, and this activation must first encounter one of those macrophages that we talked about from a second ago, right? If the antigen fits the helper T cell's antigen receptor, it can then become activated. So again, with the picture, here's the, let's say, B cell. Here's the pathogen. And I guess in blue over here, we'll do the helper T cell. Right? So here are three cells. The first thing that has to happen, let's say the first one has this going on. These little red lines. Right? The first thing that's going to happen is it's going to get ingested by the macrophage. When that happens, the macrophage will then present the antigens. And then from there, we have the helper T cell that has similar antigens that can then bind with the antigens of the B cell. Once this happens, it has activated. And now this helper T cell can do its job. Once it's activated, it can then release cytokines because it was activated. And that then can activate B cells that have already encountered the antigen. And then therefore there's more B cells and there's more people fighting off the infection. Right? So there's a lot of stuff going on here. But the point is they need to activate one another so they can do a job to help each other. Memory T cells, they don't have a delayed response like this, right? Like the last one, right? They're just ready to go. So if you have a memory T cell, these are created because there was a bacteria in the past, kind of like what we just did a minute ago. So there's some sort of bad guy that came in, right? We did this already. Let's say these are the antigens on it. And then after all the fighting is done, our body's going to produce some memory cells, some memory T cells, and they're going to remember those antigens. So when they recognize them in the future, when they bind, now they're just ready to go. They're going to send out cytokines for other T and B cells to come to the area and let's just start fighting off these pathogens because we've seen it before, and I'm ready to go. It remembers. This is called acquired immunity, right? This is stuff that you acquire over time. We can also talk about this and how vaccines are used to put people in a position to create memory T cells without actually having gotten the disease. So in this case, let's say this is polio. So if that's the case, if that's polio, then our new cells will now have that vaccine. The vaccine will give them those antigens, so it's ready to fight off a real um, polio pathogen to prevent that person from actually getting polio and letting it progress in the body. Right. So that's just one example of how memory T cells are useful for if it fought off infection already or potentially being used in a vaccine. So you don't actually have to see the real pathogen and fight it. You just get a piece of it. And then lastly, cytotoxic T cells. And these are the ones that are just going to roam around the body and they're just going to connect like we did before and destroy anything that tries to get in the body by using that perforin that we discussed in the previous video. Here's an image of everything we just talked about, so we kind of wrap it all up, right? So we have some cells that have the ability to engulf, like macrophages, right? There's also opportunities where the antigens can bind to the surface proteins of a pathogen. When they recognize them, they can release cytokines, just like you see here. So the cytokines get released. And they can send messengers to tell more cells to come in and fight off infections. So with that said, this is some information on how T cells work. And this is one of the more important lines of defense in the body. So if you have any questions on how T cells work, please let me know and we can talk about it.